Hi, my name is Jeff Groh with Calmetrix. I want to welcome you to the webinar on the study of seed germination by isothermal calorimetry. There are 5 things I want to accomplish in the webinar. First, I want to ground you in the basics of what isothermal calorimetry is all about and how it can be used to study seed germination in soil science. We'll talk a little bit about the actual instrument itself and why you might want to add one of these devices to your laboratory, and then provide you some tools for additional learning at the end of this presentation. This is a schematic of an isothermal calorimeter with the main components. We'll start on the inside of the instrument with the sample, which sits inside of a sample cup, which is 125 milliliters. We normally run this sample cup in a differential mode against an inert reference, a metal material typically, and both the sample and the reference sit on top of heat flow sensors, which are then in turn connected to a large metal heat sink. So this constitutes the actual calorimeter, which sits inside of this temperature controlled chamber illustrated in the blue. Inside of this chamber we can control the temperature very precisely from 5 to 70 degrees C. So typically in this instrument we're running in an isothermal mode. The sample is very massive, 125 milliliter, and the types of samples that we typically run in this instrument, is going to be in the order of 50 to 100 grams at least of sample, and the instrument itself is very massive. So we go to an isothermal temperature, we equilibrate there, and we look at the data as a function of time. So what's happening in the sample? There could be 2 things that might be happening. One is there could be some type of exothermic event that's giving off heat. In the case of microbiological activity or growth in a seed or whatever, there may be exothermic reactions going on within the sample. Or alternatively, there may be endothermic events in terms of the sample absorbing heat. But in the case of a sample giving off heat, basically what happens is that the heat flows from the sample into the heat sink, and the heat flow sensors can sense that. The raw signal of these heat flow sensors, which are basically thermoelectric devices, is a voltage signal, and during the manufacturing process we basically calibrate and relate that voltage signal to the heat flow in watts from the instrument. So that's basically what we're measuring, is heat flow either into or out of the sample. So the raw data, the data that we get from the instrument, is typically shown here in power and watts, though typically we will normalize that signal per gram of sample. So typically we are plotting watts per gram as a function of time. And you'll notice here that time is a very long time frame. So it's not unusual to run these experiments for 24, 48, 72, and even longer hours, and maybe sometimes in the order of 30, 40, 50 days. Just depends on the application. There's really no practical limit. There's no limit as far as the instrument is concerned in terms of the length of the experiment. It's really going to be more associated with the type of experiment that you're doing and the absolute signal relative to the baseline and so forth. So the data that we generate, we can use in a couple ways. We can use qualitatively. For instance, in a QC application, you might run a good versus bad sample, and in that context you might just be looking for qualitative differences between the curves. In other cases we might want to quantify, we might be looking for quantification in terms of the differences between the curves, the time to the various peak heights, different events that might be happening. So we use the data both qualitatively and quantitatively. I've blown up the initial portion of the curve with this instrument. Anytime you add a sample, drop a sample into the calorimeter, based on the mass and the heat capacity of the sample, you can have long equilibration times for the sample to come into precise temperature equilibration with the system. So as a result, there is heat flow generated just through this equilibration process, and that might take 2-3 hours depending on the sample. And you just have to be aware of that, both in terms of your experimental design as well as your interpretation of the data. One other thing we do with the data is we can integrate under these curves, and when you integrate power as a function of time, you get energy. So another data plot that we look at is the energy, again in joules per gram versus time. Here you see smoothly advancing curves because these are the integrals of these power curves. And again, we might use a qualitatively to compare samples, or quantification by going, say, to a specific time and looking at the total energy that is given off by the sample at a particular time. And you'll see some examples in the presentation of how we might do that. This is a good spot to point out a very important point, and that is that this technique is considered a non-specific method. And what does that mean? It means that the calorimeter doesn't really care where this heat is coming from. All it knows is that heat is being generated. And the point here is that if you have a couple different things happening within the sample, the instrument can't necessarily differentiate between the sources of those heats. So for instance, you might have a seed that has some contamination on the outside, has some disease, and as you run the experiment, you may be getting power or heat that's associated not only with the growth of the seed, but also in terms of the growth of the pathogen on the external portion of the seed. So this is important from, again, from an experimental design standpoint and from a data interpretation standpoint. One question that comes up in a lot of cases is, how does this differ with other instruments that might be in the lab, other calorimeters? And on the left is shown the typical data that we get from the isothermal calorimeter. Heat flow is a function of very long periods of time, versus another instrument that you might be more familiar with, and that's a differential scanning calorimeter, a DSC. And the point is that these instruments are very different and are used for very different purposes. The isothermal calorimeter, we use very large sample sizes in the order of tens of grams. And there's a reason that we have used very large sample sizes. One is, in many cases, we're dealing with inhomogeneous materials. And secondly, these reactions that we're looking at in these applications tend to be very small amounts of energy. So you need a large sample to get a large enough absolute signal for an instrument to be able to sense it. In addition, we use or have very long experimental times in the order of tens to hundreds of hours. Again, it's application specific because these reactions tend to be very slow and happen over very long periods of time. And then we're also interested in usually in very limited temperature ranges of, say, 5 to 70 degrees C. As opposed to DSCs, where we use very small sample sizes, typically in the order of 1 to 20 milligrams. The experiments are very short because the sample size and the mass of the system is small. We can change temperature very rapidly, which helps give additional sensitivity when we're using very small sample sizes. So these are very short experiments, 30 minutes to a couple hours long. But we're usually interested in a much broader temperature range, say minus 150 to even greater than 700 degrees C. So these instruments are used for things like looking at changes in heat capacity, glass transitions in polymers, for instance. So they're used for very different purposes compared to what we're going to talk about today, and using the isothermal calorimeter for seed germination and soil science and so forth. These kind of applications are just not appropriate for a DSC, even though both instruments basically measure power and are measuring similar types of properties. They're really designed for different applications. Baseline is very important in this instrument, as it is with many analytical instruments. The instrument is designed to provide very long-term baseline stability, which is very important to us for these applications because it basically provides a sensitivity that we need to very small changes in heat flow over very long time scales. Baseline stability is just as important in these applications as your raw signal noise in terms of being able to pick up small transitions in samples that happen over these very long time scales, and that's what the instrument's designed to do. Sample preparation is very easy. This is a picture of one of the two types of cups. These are stainless steel cups with a plastic O-ring cap that can be autoclaved. We also have plain plastic with snap lids. They're all reusable. They are very easy to load samples into because we are dealing with a fairly large cup. The main issue with sample prep is deciding whether you want to run in a closed environment where you seal the sample up and don't allow it to interact with gases other than what's in the cup, or sometimes we put holes in the cups and plumb in different types of gases, and so there's all kinds of things that we can do from a sample preparation standpoint. It mostly comes down to, how do you want to, what kind of environment do you want around your sample while you're running it. So how can the calorimeter be used in these applications? Well, as far as food science is concerned, it kind of falls into two general categories. First is this whole area of food safety and spoilage, and that includes things like looking for microbiological activity, bacteria, fungi, yeast, and so forth, and corresponding to that, the ability to preserve food, so looking at different types of preservation methods and how effective they are, estimating shelf life, and then finally looking at metabolic responses of fruits and vegetables and correlating that to things like perishability. So these are the primary things we get involved in with food safety. From a process efficiency cost standpoint, you might look at things like optimizing fermentation. Seed germination more or less falls into this category of a process efficiency. Yeast studies, optimizing the dosage of preservatives, typically in terms of trying to, not put in too much preservative, overuse the preservative, which is a cost issue. Of course, there's a lot of overlap here. You can get into issues like, for instance, in seed science where you've got surface contamination, and in that case, you really are looking at microbiological activity. So there is a lot of crossover here, but in general, seed germination kind of falls within this category. Mostly today, we're going to be talking about seed germination and soil science, and that's what we're going to focus on for this webinar. So when it comes to these kind of applications, fermentation, seed germination, optimizing preservatives, we have a lot of techniques that we use, methods that we use for studying these processes. pH meters, and we count bacteria, a lot of microscopy techniques, cell culturing, and so forth. A lot of visual type of methodologies. Many of these are qualitative type assessments, so they have a lot of disadvantages when it comes to analyzing what's going on, especially in terms of seed germination. The techniques can be very cumbersome and labor intensive. Takes a lot of training in a lot of cases. They're not continuous measurements in many cases, especially in some of the visual techniques where you might take a data point and then have to come back for some period of time later and take another data point. So they're discrete data points. Not only does that create a lot of labor requirements, for instance, if you start an experiment on Friday, you might have to have technicians or students coming in on the weekends to collect data points, but you may miss something because of the discrete nature of collecting data. So these are some disadvantages to some of the current techniques that we use, and that's really where some of the advantages of the calorimeter come in terms of, it's a quantitative methodology. It's very easy to use the technique. It's a continuous measurement, so we're collecting curves of data. You can start an experiment on Friday and come back on Monday. So it's alleviates many of the downsides to the current techniques that we use for studying these things. I will point out here that I would never position the instrument necessarily to replace some of the normal things that you've always done in your laboratory, but it's a great tool, and I think some of the applications will help you understand that, a great tool to add into your toolkit, so to speak, and augment the types of things that you're currently doing now. So with that, let's talk a little bit about germination and isothermal calorimetry, and I'll provide some background information here. Basically, you are perfectly aware of the fact that the growth of the embryonic plant in a seed is termed germination, and that process includes the absorption of water by the seed, adequate temperature and oxygen, which allows the aerobic respiration processes to begin within the seed. And of course, aerobic respiration results in the release of thermal power or heat flow, and that's what we measure directly in the calorimeter. So it's a direct measurement of the life processes that are happening within the seed as it germinates. But in addition, heat flow, as we already talked about, because of the non-specific nature of the method, can also be a result of pathogen-induced diseases on the surface of the seed. So the instrument provides a useful technique to help study surface sterilization techniques, for instance. But you do have to be aware of that and understand that in the context of designing your experiments and interpreting data. This slide, which comes from this reference here, I think is a great illustration of the fact that the process within the germination, the post-germination phase of plants is very complex. It's been studied for many, many years, and we still have very many questions on exactly what's happening. But the point is that all these processes involved with germination and post-germination, at the very beginning, respiration begins. And respiration, being an exothermic event, is something that we can sense in the calorimeter. So from the very beginning in the germination process, these processes are giving off heat, and that's what we measure in the calorimeter. So with that, let's show some examples of how the calorimeter can be used. And the first three really have to do with seeds in terms of seed longevity and storage, the effect of pulse electric field on the surface of the seeds and the impact on germination, a general application relative to assessing quality and differentiating different seed lots, and then I'll finish up with one related to really more soil science in terms of glucose-amended soil. So the first application I want to talk about is a study that was done at the Royal Botanical Gardens. You've got the reference here that you can go get that article. It's a very good article. And basically what they were interested in was to investigate calorimetry as a way to differentiate what they term good keepers and bad keepers. In other words, seeds that stored well and seeds that did not store well under certain types of conditions. So could calorimetry be used to differentiate them? This experiment, or this study, used prime and non-prime control seeds. The prime seeds were primed at 15 degrees C for 7 days using a PEG solution, and the seeds were of this variety. So after creating this two sets of seeds, the primes and the non-prime seeds, then each batch were basically divided into three and equilibrated for 2 weeks at 5 degrees C at 3 different RH levels in order for the water content within the seed to come to equilibrium within the environment. So it's 70%, 41%, 23% RH. So they ended up with basically 6 sets of seeds, one set primed at the 3 different water contents and one set non-primed at the 3 different water contents. So after the equilibration of these 6 sets of samples, then the samples were placed into the calorimeter, the temperature set at 35 degrees C, the data collected, the raw data, the power curves, collected for up to 39 days. So basically the samples were being aged in the calorimeter over this course of 39 days. And then furthermore, other sets of samples were then assessed for, based on these conditions, assessed for the percent of germination and the mean time to germinate as a way to correlate this data with the calorimetry data. So I'm not going to go into all of the data that was presented in this paper. You can certainly get a copy and read it for yourself, but I do want to point out some of the important data from the calorimeter. This data is basically there's a lot of data here, but basically I've tried to highlight the key points here. This is the data from the 70% RH water content, so based both the control seeds and the prime seeds. This panel is at the 41% RH level, and this panel at the 23% RH level. This panel happens to be a repeat of the 70% RH, but for our purposes here, I just want to highlight these two data sets. I've highlighted the curves in red and green. So you can see here, as the aging occurred, the heat flow went down. There was a difference between the control seeds and the prime seeds. You can also see a difference at the 41% RH level in terms of the relationship between the control seeds and the prime seeds, and also the absolute amount of power was different. So this is some of the raw data. They took this data and also integrated under the curves up to 6 days to create the energy curves, and basically this data is plotted at the 3 different water contents, both for the prime seeds and for the control seeds, and basically plotting the total energy up to that point, in this case up to 6 days, and plotting those 2 sets of data. So it's looking at the total heat output as a function of the seed water content and whether they were primed or control seeds. So basically there were many conclusions here, but the rate of heat flow and the total heat generated was generally greater in the control seeds, which also aged at a faster rate. The total heat generated increased with the water content. So using this data, correlating it back to the germination in the meantime to germinate, basically they concluded that it was a very useful tool as a screening tool to help assess what they call the good keepers and the bad keepers, at least at the seed lot and or species level, based on the work that they did in this particular paper. So it was basically, this example is a great example of how the calorimeter can be used to look at the conditions of storing seeds and how to use calorimetry to differentiate seeds under different conditions. Second example here is the effect of pulse electric field on the germination of barley seeds. This was a study that was done at Lund University to look at the metabolic response of germination of barley seeds after applying different levels of pulse electric field. Again, you can see the reference here and can read that paper. To begin with, the seeds were all steeped in a temperature-controlled room and allowed to germinate, and then basically samples selected based on the visual homogeneity of the germination. So they only included seeds that had actually started to germinate. And then they were basically exposed to varying degrees of pulsed electric field and then put into the calorimeter and allowed to continue to grow, basically. And the results indicate there was really no statistical difference in the metabolic heat of germination, which was being measured by the calorimeter as a function of that pulse electric field. However, there was a pretty significant reduction in the root elongation, at least at the higher levels of the PEF. So this is some of the raw data. This happens to be one of the power curves at the highest level of the PEF. But you can see the control seed versus the seeds that were exposed to these high levels of pulsed electric field did not really vary. Of course, in this case, the power curves are going up because we're actually now in a growth mode where there is germination going on, or plant growth going on. So the power curves are going up versus in the seed storage situation where the seeds were not germinating, they were aging. The power levels were going down. So they took this raw data and basically integrated under these curves at the various conditions of the electric field, and so that's what this data is. It's individual discrete data points, the individual energy associated with the individual electric field. So you see individual data points here as a function of the electric field and the total heat flow, or the total power, I'm sorry. And you can see that as a function of the electric field, it's pretty constant. So that's where their conclusion came, that the gross metabolic processes were the same as a function of being exposed to the electric field. However, the root length did decrease as a function of this electric field. So this is another great example of how calorimetry can be used to assess different processing conditions on the seeds and the metabolism going on in the seeds versus their root length in this case. So it's a very powerful example of that. The third study is really one that was done to illustrate the use of the instrument to assess quality of seed lots. Again, these were barley seeds. There were 5 kind of things, and I'm going to highlight the use of the technique to look at the effect of sterilization, differentiating different seed varieties, how reproducible the data is, just a difference between what you would see for seeds that have already sprouted versus normal seeds, and the differences again in aged versus unaged seeds. In this particular study, barley seeds were used. It was actually determined that 10 seeds was appropriate, was plenty in order to get a big enough signal, but 50 were used. They were introduced into the base of this plain plastic sample cup. You remember I showed you a picture of the stainless steel cup. These are the other types of sample cups. They're just a plain plastic cup with a snap lid. You're looking down into the sample cup. So the seeds were placed on filter paper that was soaked in water and then preconditioned at 20 degrees C. So these were then put into the calorimeter, and the data recorded as a function of time as the seeds germinated and began to grow. So let's look at some of the data. One of the questions that comes up from time to time is back to this issue related to surface contamination. So in this experiment, the regular seeds were run, and then some seeds were sterilized and also run. So the point here is, by looking at this data, the conclusion was there was not significant pathogens on the external portions of those seeds. There was not significant enough difference between the sterilized and the non-sterilized seeds to indicate that there was a problem associated with disease on any of the seeds. So that's one use for the instrument, is to look at surface contamination and pathogens on the surface of the seeds. This is an example where you see the signature basically in a qualitative way between 2 different varieties of seeds. So, you know, every seed in every variety will typically have a signature curve, and so you can use it in that way to differentiate between different varieties. One question that comes up is reproducibility. So this is 4 sets of data, and actually this has been normalized on active seeds. So if all the seeds were included, there would be some variation in data, but once you remove the seeds that had not germinated and normalize on that weight, then you get very good reproducibility across 4 sets of data. So again, this gets back to understanding what the instrument's measuring, how you're treating the data, and making sure that you know, you're making apples-to-apples comparisons. This is just an illustration of seeds that were already sprouted versus regular seeds, and again, the difference between the seeds that have already sprouted are generating a lot more energy over time compared to the normal seeds. And then finally, some seeds were aged for 7 days in a furnace, and then put into the instrument and run against the control seeds. And you can see the dramatic difference between the growth pattern here, the heat signatures here of the seeds that were aged and the seeds that were not aged. And there's lots of data out there that collaborates this conclusion that the aged seeds, you know, will show this type of signature compared to seeds that have not been aged. So again, this is a great example of how the calorimeter can be used in a variety of applications for looking at seeds. The final application here is really related to soil science. Excuse me. So in this case, the sample cups were both filled with water initially and allowed to come to equilibrium for 8 hours. So you can see the data equilibrates here at 0. There's nothing that should be going on. Once the water is equilibrated with the environment, you should be basically at zero thermal power. And then soil was introduced into both cups and allowed to come to equilibrium. Again, there are processes going on as soon as the soil was introduced, and so there are constant exothermic events that are occurring in the sample. At that point, one sample, some additional water was added, that's the green curve, and the other sample, a glucose solution was added to the sample. And you can see, of course, that's a food source, and now there are other activities going on within the soil. So by integrating under this curve, you could look at things like biomass conversion and other useful pieces of data from this experiment. So this is just an example of how the calorimeter might be used in a very common experiment in terms of glucose-amended soil experiments. Okay, so hopefully those 4 examples provided you a really good background on how calorimetry is really just an excellent tool in the seed, in the seed science and soil science area, in terms of looking at seed germination under a variety of conditions, the ability to differentiate aged versus unaged seeds, the concept of seed longevity and how seeds age under various different storage conditions, the effect of sterilization techniques, differentiating diseased versus healthy seeds, and all the things that you can do relative to soil science. So just a plethora of different types of applications. And really, one of the points I want to make is that the calorimeter is very flexible. Really, the applications are only limited by your imagination. If you think about that sample cup, it's a large 125 milliliter sample cup. There's a lot of things that you can do creatively in terms of sample prep and so forth, and the instrument being general in nature in terms of measuring heat. Like I said, the applications really are only limited by your imagination. So now let's talk a little bit about the instruments. There are 2 different versions, the 2000 and the 4000. These are all the different applications, some of which we talked about today. Both instruments can basically run anything. There are differences, however. The 2000 has the ability to run 2 samples simultaneously, and the 4000 can run 4 samples simultaneously. So this is the highest throughput instrument. There are other differences. The 2000 is really considered our highest performance calorimeter. It uses the 225 milliliter sample cells. The sample cups are the same between the 2 different instruments. There's no difference there. Temperature range of 5 to 70, again, that's the same between the 2 instruments. The duration of test here I've indicated as hours to days. Most of the experiments that we do might be 24, 70, 48, 72 hours, 2 to 3 days, but it doesn't preclude you from running 30, 40, 50 days. It really just depends on the application and what you're trying to measure. And both instruments can run in those modes. The 2000 is our highest performance instrument. It has the best baseline performance, so it has the maximum sensitivity to picking up the smallest amount of change in the baseline, basically. It's optimized for minimum crosstalk because it uses 2 individual calorimeters with completely separate cells, so that's why this is the highest performance instrument. The instrument comes with the software. It comes with actually a computer. It's a turnkey system. You can get this system, get it out of the box, get it up on the bench, and within a half an hour you're ready to go, or you're at least got the instrument equilibrating. Very easy to install and use the instrument. The 4000 is considered our high performance calorimeter. Uses the same 125 milliliter sample cells and sample cups, but it has 4 samples, so you can run 4 simultaneously. So it has twice the throughput as the 2000, which might be important in many cases. Temperature range the same duration of test. Very excellent baseline performance. In most situations that we're dealing with, it's going to be perfectly suitable. In terms of the application. It includes the software and computer, just like the 2000. So those are the 2 different instruments that are available. Both instruments come with the software. It's very straightforward, easy to use software. It's a single application. There's one tab for the data analysis portion that allows you to open up the 8 data files and overlay those and analyze the data. There's another tab here, the BioCal logger, which is basically the instrument control portion of the software. Software is very straightforward, very easy to use, and included with the instrument along with the computer. So hopefully, now the question is, why might you add this instrument to your lab? And hopefully I've given you some powerful reasons through the applications. This technique is a proven, powerful analytical technique. While it might be new to you and it might be new to the seed science and soil science area, the technique itself has been around for more than 30 years, and it's used in a wide range of different applications. This instrument is really optimized for food science and the types of things that we're doing here, but the basic concept has been around for many years. It's very easy to use. I can train anybody to use this instrument from a technician level up to a high-level PhD type person. So it's very easy to use, broadly applicable, because it's directly measuring heat, and that's what all food science is basically, is life processes. It's broadly applicable to many different situations. It's very robust. I have no qualms at all putting this instrument into a academic laboratory where there are a lot of undergraduate or graduate students using it. It's very robust compared to a lot of techniques, especially when you compare it to things like microscopy and cell culturing and some of the other equipment that you need for some of these other techniques. It's very robust. And it's very cost effective. High value in use. For the amount of money that you will spend to buy this instrument, it's very cost effective. And so, it has a high value in use, type of value proposition. It's applicable to both R&D situations and QC. It's perfectly suitable in either situation. You can save a lot of time and money with the instrument while simultaneously expanding your testing matrix, which gets you data much faster. So you can do a much bigger test matrix. You can run multiple parallel experiments. So, and the fact that it's a continuous measurement, you can start an experiment on Friday, you come back on Monday, you've got data without having to bring people in over the weekend. So many, many reasons why this instrument can save you time and money and get you higher quality and more data faster. In many cases, it can reduce your laboratory capital costs because you don't have to have as much expensive other equipment. Reduces your overall equipment capital cost. It uses very little bench space compared to other techniques, so you have much more higher throughput, more data per linear foot of bench space, so to speak. So many reasons why it can reduce your capital cost requirements. And finally, we have on staff some of the world-renowned experts in calorimetry. They invented this technique more than 30 years ago. Anyone can sell you an instrument, but the staff that we have available, the expertise that we have, can help you get the benefit out of the instrument, and that's really important and really why we focus on providing that outstanding customer support through the expertise that we bring to bear on these applications. Okay, so I'm going to finish up here. There are many things that you can get from visiting our website in terms of videos, application articles that are available for download. I also have access to many others based on your specific application. So you can visit the website. We offer on-site seminars. These are usually done in the context of an instrument purchase in order to provide advanced training. We have a very nice rental program. The instrument itself is more difficult to demonstrate on site because it does take time for the instrument to equilibrate to the lab temperature and be ready to run experiments. And then the experiments themselves, take 24, 48, 72 hours at least. So we do have a very nice rental program that allows instruments to be put into labs for 1, 2, 3 months and an application assessed. It's very cost effective. You pay a nominal monthly fee, and all those rental fees can apply to the purchase of the instrument. So it's a great way, in a very cost effective way, to assess the instrument and then put it into your laboratory, or add additional capability in your lab if you've got a big project that's coming up and you need more capacity. We can do sample analysis under certain conditions. It really depends on the application, the sample, what kind of sample prep issues there are, and so I encourage you to get in contact with me, and we can talk specifically about your application and what the best way might be to handle it. So I'll finish up here with my contact information. I'm very happy to answer any questions for you, and want to thank you for attending the webinar today, and hope you found it informative and useful. Thank you.